Looking closely at a human brain undergoing severe neurodegeneration, we observe specific physical hallmarks. There is localized neuronal loss, tau hyperphosphorylation, and the aggressive aggregation of misfolded alpha-synuclein proteins, destroying neural tissue. For decades, medical science classified conditions like Alzheimer's and Parkinson's strictly as central nervous system diseases. But recent autopsies and in vivo data present a glaring anomaly. Deep inside these diseased brain tissues, researchers are finding lipopolysaccharides, or LPS, bacterial endotoxins that originate exclusively in the gut. The concentration of these peripheral bacterial byproducts is not random. It scales directly with the severity of a patient's cognitive decline. To trace the true origin of this neurodegeneration, we have to look past the skull and remap the anatomical barriers and communication circuits of the gut-brain axis. This diagram maps the digestive system, a continuous tube from the oral cavity through the gastrointestinal tract. Connecting this tract to the brainstem is the vagus nerve. Alongside the vascular network, it forms a bidirectional communication circuit between the gut and brain. The gastrointestinal tract operates as a highly innervated sensory interface, constantly monitoring its internal chemical environment and feeding that data back to the brain. When the microbial populations residing inside these digestive spaces fall into a pathological imbalance, a state known as dysbiosis, this sensory interface becomes a systemic vulnerability. Microbial dysbiosis leverages these exact anatomical highways. The body's own digestive network turns against the brain, actively driving central neurodegeneration. This cross-section illustrates the histological layers of the gastrointestinal tract. The innermost layer, the tunica mucosa, composed of epithelial cells, lamina propria, and muscularis mucosa, acts as a selectively permeable blood-gut barrier. Embedded within this mucosal epithelium are enteroendocrine cells, or EECs. These sensory cells continuously detect changes as luminal nutrients and microbial metabolites flow past them. When triggered, EECs initiate a neural handshake. They emit signaling molecules, specifically using the neurotransmitter glutamate, to communicate directly with vagal afferent fibers. These vagal nerve endings sit just beneath the mucosal boundary, waiting in the tissue layer known as the tella submucosa. In Parkinson's disease, the pathological alpha-synuclein protein actually begins accumulating here in the gut long before it appears in the brain. This schematic demonstrates retrograde transport. Misfolded alpha-synuclein molecules enter the peripheral nerve terminals of the vagus nerve and travel sequentially up the axon, propagating directly into the brainstem. Evolution designed this neural network to transmit satiety and nutrient data. Instead, it is actively hijacked, serving as a physical conduit for neurodegenerative proteins to reach the central nervous system. Zooming in on the tunica mucosa in this diagram, we see the projecting microvilli, or brush border, sitting directly above the red and blue vascular networks of the lamina propria. In a healthy state, commensal bacteria above these microvilli ferment complex carbohydrates into short-chain fatty acids, or SCFAs. One key SCFA, butyric acid, crosses the epithelial barrier and enters underlying blood vessels to join systemic circulation. In the bloodstream, SCFAs perform critical functions. They stimulate regulatory T cells to balance immune responses and maintain the strict integrity of the blood-brain barrier. Ultimately, these circulating SCFAs reach the brain, where they suppress the activity of microglia, keeping neuroinflammation in check. This split screen models the shift to a dysbiotic state. As pathological strains overpopulate, protective butyrate drops, and bacteria shed toxic lipopolysaccharides, or LPS. The tunica mucosa degrades, creating a leaky gut that allows LPS molecules to flood into the underlying vascular layer. Once systemic, this LPS circulation bypasses the compromised blood-brain barrier. Upon entering neural tissue, LPS violently activates microglia through toll-like receptors, triggering an aggressive immune response. Grounded, the physical integrity of the tunica mucosa is the precise tipping point. Its failure dictates the shift from systemic neuroprotection to chronic neurodegeneration. Returning to our anatomical map, we move up from the lower GI tract to the oral cavity, which harbors the body's second largest microbial community. This region presents a critical vulnerability. The periodontal route allows pathogens to completely bypass the complex, multi-layered barriers of the lower GI tract. Observing periodontal tissue, oral pathogens actively secrete proteolytic enzymes that dissolve local connective tissue fibers, allowing them to slip directly into the bloodstream. Forensic pathology provides proof of this infiltration. Researchers have isolated toxic proteases called gingerpains, produced specifically by P. gingivalis, inside the post-mortem brains of Alzheimer's patients. Once these oral bacteria and their enzymes reach the brain, they inflict severe localized damage. They disrupt axonal transport, alter mitochondrial migration, and initiate neuronal apoptosis. Whether the dysbiosis occurs deep in the gut or in the shallow tissues of the gums, these peripheral mucosal infections possess the exact biological mechanisms required to directly seed central neuroinflammation. Summarizing, this schematic isolates the three precise pathways connecting the peripheral microbiome to the brain. There is the neural route via the vagus nerve, the chemical route driven by microbial metabolites, and the systemic immune route caused by vascular leakage. The clinical data confirms that severe central neurodegeneration is rarely an isolated event in the brain. It is the final cascade resulting from a physical failure at the body's mucosal interfaces. To protect the brain, medical science must shift its therapeutic target. Intervention has to focus on the peripheral source of the inflammation. This has led to the development of targeted psychobiotics. These specific bacterial strains are supplemented to therapeutically alter and correct the composition of the gut microflora. By doing so, psychobiotics work to physically repair the intestinal barrier, sealing the leaks and artificially restoring high levels of neuroprotective SCFA production. Alongside chemical interventions, physicians are utilizing mechanical methods like vagus nerve stimulation, or VNS, to manually send electrical signals along the gut-brain axis. VNS drives specific anti-inflammatory cholinergic pathways. This peripheral stimulation helps maintain the integrity of the blood-brain barrier and actively inhibits microglia activation in the central nervous system. Treating and potentially reversing cognitive decline requires a direct shift in focus toward diagnosing and healing the microscopic ecosystems of the digestive tract.